All right, the purpose of this video is to give students an opportunity to prepare for the genetics test, which will be done in the afternoon sessions this Friday. For those people that are in the meeting, they heard me say this, but there are items on your study guide. There are items in this Jeopardy that are not actually going to be tested on. The study guide was provided for the students prior to us learning that we were going to go into the hybrid. And of course, that meant we went from an hour and a half in our classes to 55 minutes. So I had to shorten the test to make sure that students have a good opportunity to do their best on that test. So I think that there are 34 multiple choice questions on the test. The study guide in this Jeopardy game represented, I believe, 40 previous multiple choice questions. There are a couple extra genetics problems that are no longer on the test, and there are some essay questions that used to be on the test that are no longer on the test. So that's how I shortened it, by removing those questions, but I'm not going to go into which questions I removed. As long as you understand and know everything on the study guide, you will hammer this test. All right, so here we do. We're going to go one column at a time. I'm going to give you about, 2, 5, 9, 14, 17, 19, 27, 29, 32, 36, 41, 44, 44 and 6, 47, 49. So we'll do about 1 minute per 100-point question. So about 2 minutes for the 2-pointers, 3 minutes for the 3-pointers, and 4 minutes for the 4-pointers. That may be more time than you need, but that's just fine. All right, here we go. All right, genes that mask or are stronger than others are said to be blank. If two plants are big T and big T little t, they will have the same what? Okay, a couple more seconds. All right, here we go. All right, they are said to be dominant over the recessive alleles or the recessive genes. And of course, as long as you have one dominant allele, you will have the same phenotype. A capital R represents, hold on, let me get my timer going. A dominant allele, and a lowercase r represents a recessive allele. What would the genotype of the hybrid be? The principle of blank states that some alleles are dominant and some are recessive. The genetic makeup of an organism is called its blank. Okay, and again, you probably know the answers to these questions. That's just fine. Just use the extra time as an opportunity to review your study guide. All right, a couple more seconds. All right, here we go. Okay, so a hybrid has characteristics of both parents, and a hybrid is heterozygous. Okay. Principle of dominance states that some alleles are dominant and others are recessive. And of course, the genetic makeup or the symbols that represent a trait are the genotypes. The easiest way to remember the genetic makeup is the genotype, the physical appearance is the phenotype. All right, the chemical factors that determine traits are called blanks. The symbols or letters used to represent a recessive trait is or are what? So is it singular? Is it plural? Which scientists used hybridization to make a new line of plants? And traits that are produced by the interaction of several genes are called blank. And during the silence, you guys, if you have any questions, just unmute yourself and ask away. I'd be happy to talk to you about other things, even other than these questions, while we're waiting. But do understand your questions and the answers will be on the video that potentially other students will be watching. So understand that. All right, a couple more seconds. All right, here we go. Okay, so genes are the chemical factors of traits. Two lowercase letters represent a recessive trait, so that's going to be homozygous recessive. Luther Burbank, of which a high school in the Sacramento Unified School District is named after. Luther Burbank used hybridization to make a new line of plants, and traits that are produced by the interaction of several genes are referred to as polygenic, polymany-genic genes. Here we go. Let's get into some genetics problems. Pea plants have what? Sorry about that. Let me correct that. two different alleles, sorry. If pea plants have two different alleles for the same trait, they are said to be blank. Being tall is dominant to being short. A man heterozygous for tall and a short woman have children. What is the probability in a fraction that their kids will be short? You will have to do a one trait cross problem. You're going to have a blood type problem. You will have a codominance problem, and you will have an incomplete dominance problem. I don't think that there is a two trait cross problem on this test. There certainly is not a sex-linked trait problem on this test. And speaking of which, in the Google Classroom, if any of you guys are interested in seeing how a sex-linked trait problem is worked, I worked 3 of them, and I put a video in Google Classroom as material. It is not homework. It is totally optional. But if you are curious about how those problems are worked, it is there for you. All right, about 30 more seconds. All right, here we go. All right, so if they have two different alleles, sorry, I'm a perfectionist when it comes to this stuff. I forgot to correct it here too. All right, two different alleles for the same trait, they are heterozygous. Being tall is dominant to being short. Man heterozygous for tall, and a short woman have children with the probability in the fraction that their kids will be short. That is 1/2. It would be 50%. Would be a 1-to-1 ratio of being tall to short. It would be a 1-to-1 ratio of, hold on, heterozygous, big T, little t. It'd be a 1-to-1 ratio of big T, little t to little t, little t. So those are the only 4 types of questions I could ask you about that problem. All right, here we go. A heterozygous plant and a homozygous recessive small plant are crossed. What's the probability in a percentage that the dominant trait will be seen in the offspring? Sketch a chromosome and label both the centromere and the chromatids. This is one of the questions that is not on the test. I tested you guys on meiosis and mitosis and chromosomes on the last test. Therefore, there is no need for you to sketch the chromosome and label the parts here. You can still do it, but you don't need to do it. Okay, a couple more seconds. All right, here we go. Okay, so it's 50%, and of course, the centromere is the middle region, and the sister chromatids are these 2 pieces here. But again, that question is not on your Friday test. All right, using the labels of different dominant and recessive alleles to explain how crossing over leads to more genetic variation. This is not on the test, but I will show you how that would be described. So with no crossing over, gametes would only have the AB. Let me move this out of the way. The big A, big B combination, all right, because of these two here, and little a, little b combination. When crossing over, gametes would have a big A, little b, or a little a, big B combinations, and different allele combinations lead to different characteristics in offspring, which can lead to genetic variation and can lead to evolution of that species. So genetic variation strengthens species. All right, here we go. We'll get back to the genetics problems a little later. Let's get into Mendelian genetics, simple genetics. Homologous chromosomes, sex chromosomes, and autosomes are all shown in a blank. Gregor Mendel used pea plants to show the blank of traits or characteristics. All right, here we go. Okay, so those are all found in a picture of the chromosome pairs, and that's called a karyotype. And Mendel used pea plants to show the inheritance of traits or characteristics. Okay, let me get my timer going. All right, blank is the likelihood that a particular event will occur. The visible or physical characteristics of an organism are called blank. An organism that has the same genes for a particular trait, like big A, or little a, is said to be what? Okay, a couple more seconds. All right, here we go. Probability is the likelihood that a particular event will occur or not occur. Visible or physical characteristics are the phenotypes, and if they have two of the same allele, they are referred to as homozygous. All right, here we go. Units of heredity are called blanks. True or false? Mendel used pea plants because they only have a single visible trait, which makes them easier to study. When Mendel crossed true reading tall and short plants, all the plants were blank because the allele for blank plants is dominant. If a Punnett square has big T and big T little t, the offspring will be, and you can give me the phenotype or the genotype for that last question. Okay, about another minute. All right, here we go. Units of heredity are called traits. There were a number of different traits in pea plants that Mendel was studying. So that is a false statement. Let's see, when Mendel crossed true breeding tall and short plants, you got only tall because the tall allele was in all of the heterozygotes or all of the hybrids. Therefore, in the F1 generation, all of those organisms were tall. Let's see, if a Punnett square has big T, and big T, little t, all of the offspring are going to be tall because all of the offspring will have at least one uppercase T. You would have two heterozygotes and you would have two that are big T, but they still would all be tall. All right, when black and white parent chickens produce a speckled chick, this is an example of blank. Human skin color differences are an example of blank. All right, about 30 seconds. Okay, here we go. All right, so it's codominance because both dominant traits are actually seen in a speckled chick. And polygenic trait is human skin color, human hair color. What was the other one? Eye color, okay, influenced by many genes. Okay, true or false? Oh, these are different. Okay, so this is B-type blood. So just look at the B and the B, ignore the I's. I forgot to go in here and change these. And then this is going to be capital B, O. All right. So BB and BO both result in the same phenotype. True or false? Most sex-linked traits are found on the blank chromosome. That should sound kind of familiar. How many pairs of chromosomes are found in humans? Okay, 30 seconds. All right, here we go. Okay, so that's true. Capital B, and capital BO, both of them result in B blood. Of course, this one would be homozygous dominant, and this one would be heterozygous, but because the B allele is dominant, you'll see it, B blood, both cases. Most X-linked traits are on the X chromosome. There are a few on the Y, but most are the X, and 23 pairs of chromosomes in humans. All right, here we go. If a couple has 3 boys in a row, what is the probability that their next child will be a boy? Compare and contrast Turner's and Klinefelter's syndromes. And I'm sorry if this audio is picking up my CD player. I'm in the process of burning a couple CDs. Sorry if that's annoying to anybody. All right, about 30 seconds. All right, here we go. Okay, so regardless, it's going to be 1/2. The next child is always going to be 1/2. Now, if I were to say what is the probability that you have 4 straight boys or 4 straight girls, that's different. then you'd have to multiply a half times a half times a half times a half. And of course, the probability of that happening would be much less than 1/2. But if we're just talking about a single event, it's always 50-50. Okay. All right. So similarities between Klinefelters and Turners, they are both caused by non-disjunction. If you're not familiar with what that is, you probably haven't done the 14-2 class notes yet, but that's basically during meiosis 1, an anaphase, when the chromosomes are supposed to be pulled apart from one another, that fails. Both chromosomes end up in one of the daughter cells, and of course the other daughter cell would have none of the chromosomes, and if that egg cell or sperm cell with too many or too few chromosomes ends up in the fertilized egg, then you have a problem. Okay, so they're both caused by non-disjunction. They both cause sterility. They both cause developmental and learning problems. They both result in anxiety. And differences, Turner's is in females. Klinefelter's is in males. Turner's has 1 less X. Klinefelter's has 1 more X. Okay. Okay, in the P generation, a tall plant, hold on, let me get the timer going. A tall plant was crossed with a short plant. Short plants reappeared in the F2 generation because the allele for shortness and the allele for tallness blanked when the F1 plants produced gametes. Down syndrome is also referred to as blank 21. That term for Down syndrome is also in the 14-2 class notes. So if you haven't done those reading notes yet on that section, or if you haven't done those class notes, you may not know what the answer to this second one is. Of course, for all of you who are watching this Wednesday evening or Thursday or Friday sometime, I'm hoping that you did the 14-2 class notes and that you did them on time. All right, a couple more seconds. All right, here we go. Okay, so they get separated or segregated is the best word, and trisomy basically means 3 of chromosome number 21. So what that meant is that it got an extra chromosome, which means that either the sperm or the egg from the mother or the father had 2 copies of chromosome 21, and then when you added it during fertilization, that gave it 3 copies. All right. Okay, the law of blank states that 1 gene or allele from each pair goes to each sex cell. If a man heterozygous for dominant brown hair and a woman with blonde hair have kids, what would the phenotypic ratio be, or the phenotype ratio be? The principles of blank can be used to predict the traits of offspring produced by genetic crosses. And a blank blank shows all the possible results of a genetic cross, the genotypes of the offspring, and alleles in the gametes. Doing a video. Couple more seconds. All right, here we go. The law of segregation says that each gamete gets only one copy of the gene. The phenotypic ratio is going to be 1-to-1 or 2-to-2, brown hair to blonde hair. Of course, if you ever have a 2-to-2, a 3-to-3, a 4-to-4, it should be reduced to a 1-to-1. Let's see. Principles of probability can be used to predict offspring produced by genetic crosses, and a Punnett square shows all possible results of a genetic cross, the genotypes of the offspring, and the alleles, or the letters, that come in the gametes. All right. Alyssa and Mike, her brother, have brown hair, dominant trait. Alyssa's son does not. Draw a pedigree chart. And what is the husband's genotype? What are the 23rd pairs of chromosomes in human males and females called? You will not have to draw a Punnett square for the test. I'm pretty sure you will be given one and you'll have to answer questions about it. About 30 more seconds. Okay, here we go. All right. So the last pair of chromosomes, they're called the sex chromosomes. Okay. So this is kind of a tricky question because we could not tell if it was homozygous dominant or heterozygous, the son-in-law, or beg your pardon, the father that was marrying into the family. Pardon me. This is Alyssa's brother, Michael. Alyssa is heterozygous. Her husband is unknown. And then, of course, the son is going to be homozygous recessive. All right, some genetics problems. I'm not sure how long you guys will have or how long it'll take. You can skip a couple steps if you need. Oh, don't worry about sex-linked trait problem. Yeah, okay, I'm not going to worry about that one. Okay, a woman with AB blood and a man homozygous for A blood have children. What are the possible phenotypes of their children? What is the genotype of the woman? And what are the two genotypes for A type blood? All right, about 15 more seconds. All right, here we go. Okay, so what are the possible phenotypes of the children? It's going to be A, B, and A. Remember, ignore the I's. So the second question, what is the genotype of the woman? She is A, B. All right. What are the two genotypes for A-type blood? It's going to be A, and A, O. The little I is recessive and stands for the O allele. Rolling tongue is dominant to not being able to. A woman heterozygous and a dad who cannot roll his tongue have kids. What is the phenotype ratio expected in their children? White and red flowers are incompletely dominant to one another. Red flowers crossed with a pink one. What is the genotype ratio expected in the offspring? Okay, about a minute and a half left. Okay, 30 seconds. All right, here we go. Okay, so first question, what is the phenotype ratio expected in the offspring? That's going to be a 1-to-1 or 2 to 2. 2 to 2 should always be reduced to 1 to 1. Rolling the tongue to not being able to. Second question, what is the genotype ratio expected in the offspring? That's going to be 1 to 1, big R, which is going to be red, and big R, big W, which is going to be pink. All right, ladies and gentlemen, that is it for all the questions. Thanks for being here. Of course, the test will be on Friday in the afternoon, regardless of whether you're in cohort A, B, or C. Please get in touch with me if that time is not going to work for you. We'll see y'all later.
Thanks, Mr. Collins.
You are welcome.